Welcome to the deep dive. Today, we're really zeroing in, taking a very close look at human genetics. We're focusing on just one specific spot on a chromosome, a spot that, well, when it's missing, it fundamentally changes a person's entire physical and neurological development.
Yeah, that's right.
We're dedicating this deep dive to chromosome 22q11.2 Deletion Syndrome.
And it's a fascinating area. This deletion, it's surprisingly common, relatively speaking, for a chromosomal disorder.
How common are we talking?
Well, figures suggest maybe as high as 1 in 4,000 live births. But the really interesting part, historically anyway, is how studying it forced a major rethink.
Or rethink how?
It made the medical community realize that two conditions, previously thought to be totally separate syndromes, were actually just different faces of the exact same underlying genetic issue.
Ah, okay. So this discovery kind of unified the field in a way.
Absolutely. That structural reorganization of knowledge was huge.
So our mission today is to really unpack the details of this deletion using the pathology sources we've got. We want to understand how losing this, well, tiny piece of DNA can cause such an incredibly broad range of problems.
And critically explore its connection to things like adult psychiatric illness, which is a major aspect.
Definitely. Let's start with the basics, the name itself, 22q11.2. Can you break down that notation for us? It sounds like coordinates on a map.
It essentially is. It's the cytogenetic notation, the language used to map chromosomes visually under a microscope. So 22 is simply chromosome number 22.
Okay.
And each chromosome has two arms, a short one P for petit and a long one Q.
So Q means we're on a long arm. Got it. What about the numbers, 11.2?
Those pinpoint the location more precisely. Think of it like region, band, and subband. So Q11.2 means long arm Q, region 1, band 1, subband 2.
So it's specifics.
Very specific spot. And it's within this tiny band that a segment of DNA is missing or deleted. We're talking about roughly 1.5 million base pairs.
And the absence of that specific segment kicks off this whole cascade of issues across multiple body systems.
That's the one.
You mentioned the historical significance, the merging of conditions. That must have been a real landmark discovery.
Oh, it was revolutionary. For a long time, clinicians knew about DiGeorge Syndrome and velocardiofacial syndrome, or VCFS. They saw some overlap, sure, but they had enough distinct features that they were treated as separate entities.
Right.
The breakthrough came when researchers found the exact same deletion, 22q11.2, in patients diagnosed with both conditions.
The underlying cause was identical.
Identical, and suddenly the picture snapped into focus. It wasn't two syndromes, it was one syndrome with a very wide spectrum of possible symptoms.
So chromosome 22q11.2 deletion syndrome became the umbrella term?
Precisely. And that huge variability in how it presents, the phenotype, it makes sense when you realize what's lost. It's not just one gene malfunctioning.
No, you said it was quite a few.
It's about 30 to 40 genes packed into that deleted segment. Losing half the normal dose of instructions from that many genes, well, it can impact almost everything. The heart, the palate, immune function, how the body handles calcium, even cognitive abilities.
That really does set the stage for complexity. And if it's, as common as 1 in 4,000, but the features vary so much.
Exactly. It makes sense why cases might be missed or maybe diagnosed late, or perhaps only one symptom, like a heart defect, gets noticed initially.
Right, without seeing the whole picture.
Correct. That lack of a single uniform presentation makes detection a real challenge. It underscores why clinicians need a deep understanding of the full possible spectrum.
Okay, so to really get a handle on that spectrum, let's dig into those historical definitions first. Why did DiGeorge and VCFS seem so different initially?
That's a great question. It mostly boils down to which symptoms were most prominent and often when they appeared. Hashtag Technico, historical syndromes. Under the 22q11.2 umbrella, let's take DiGeorge syndrome first. The classical description really focused heavily on the immune system and the endocrine system, particularly calcium regulation, and it often presented right at birth, sometimes as a crisis.
What were the absolute key features for that classic DiGeorge picture?
The core issues stem from problems during embryonic development, specifically with structures called the third and fourth pharyngeal pouches. Problems here lead directly to the main DiGeorge features. First, thymic hypoplasia.
Hypoplasia meaning underdeveloped.
Exactly. The thymus, which is where crucial immune cells called T cells mature, is too small or sometimes even absent. This leads directly to T cell immunodeficiency.
And T cells they're like the conductors of the immune orchestra, right?
A good way to put it. They coordinate much of the adaptive immune response. Without enough mature T cells, the body's defense against many viruses, fungi, and certain bacteria is severely weakened.
So a life-threatening vulnerability from the start.
Potentially, yes. It can be very severe, needing immediate medical care in infancy. Sometimes things like thymus transplantation are considered.
Wow. Okay, what was the second key component of DiGeorge?
Parathyroid hypoplasia. The parathyroid glands, which control calcium levels, also develop from those same pharyngeal pouches. If they're underdeveloped, they can't produce enough parathyroid hormone.
Leading to low calcium.
Right, hypocalcemia. Dangerously low calcium.
And what does that look like functionally in a baby?
Low calcium makes nerves and muscles overly excitable. So you can see things like muscle spasms, twitching, or even seizures, which are obviously medical emergencies.
So severe immune issues and calcium problems right out of the gate.
That combination really defined the acute, often neonatal presentation of classic DiGeorge. There were often associated heart defects too, specifically affecting the outflow tracts, and sometimes mild facial differences.
Okay, now let's contrast that with the velocardiofacial syndrome, or VCFS. You said it sounded less like an immediate crisis.
Generally, yes. VCFS, at first glance, seemed more focused on, well, structure and cognition. The name itself gives it away. Velo for the soft palate, cardio for the heart, and facial for the face.
Makes sense. What were the defining signs for VCFS then?
Key features included facial dysmorphism. Things like a prominent nose, maybe underdeveloped cheekbones, a small or recessed jaw that's called retrognathia, and very characteristic were palate problems.
Like a cleft palate.
Often, yes. Either a visible cleft palate, a hole in the roof of the mouth, or sometimes a submucous cleft, which is harder to see but still affects function. These palate issues really impact speech, causing a hypernasal sound.
And the cognitive aspect you mentioned, how did that differ from DiGeorge?
This was a major point of distinction initially. While babies with DiGeorge often had developmental delays, individuals identified with VCFS typically showed clear learning disabilities. And, importantly, they seemed to have higher rates of behavioral issues and later, psychiatric problems. Diagnoses might come later in childhood because of these aspects.
So immunodeficiency was less of a focus in VCFS.
It could still be present, but usually not as severe as in the classic DiGeorge presentation.
It really is striking that one deletion could cause such different primary problems. Severe immune deficiency versus, say, major speech and learning issues.
It is. But the crucial overlap, the thing that eventually connected them, was the presence of heart defects and the facial or palate abnormalities in both groups.
That was the clue pointing to the same underlying genetic spot.
Exactly. That shared ground strongly suggested a single cause. And it's worth mentioning, as the sources note, while 22q11.2 is overwhelmingly the cause, a very, very small number of cases that look similar might involve a deletion on chromosome 10, specifically 10p13.14.
Ah, so there can be rare mimics.
Genetics always keeps you on your toes. But for all practical purposes, when we talk about this constellation of symptoms, we're focused on 22q11.2, ##B, major clinical manifestations in depth.
All right, let's dive deeper into the specific systems affected, starting with the heart. You called cardiac defects the survival challenge. Why so critical?
Because they're often the most immediate threat to life, especially in newborns. The deletion specifically messes with the development of the heart's major plumbing, the large vessels carrying blood away from the heart. We call these conotruncal defects, or outflow tract defects.
Okay, outflow tract. And what specific heart conditions are most common with this deletion?
Two big ones stand out. Tetralogy often abbreviated TOF, and truncus arteriosus.
Tetralogy of Fallot. That involves multiple issues at once, right?
Yes, tetra means 4. It's a complex defect, including things like a hole between the ventricles and narrowing of the valve leading to the lungs. Truncus arteriosus is arguably even more complex. Instead of two separate main arteries leaving the heart, the aorta for the body, and the pulmonary artery for the lungs, there's just one single large vessel, or trunk.
Which means oxygenated and deoxygenated blood get mixed right away.
Precisely. Both conditions require complex surgical repair, often very early in life.
And the link is strong. I remember a statistic about this.
Yes, it's quite striking. About 30%, nearly a third of all individuals born with one of these isolated conotruncal heart defects, even if they don't show the other typical DiGeorge or VCFS features, actually have the 22q11.2 deletion when tested.
Wow. So this deletion is a major player in causing these specific severe heart problems.
A very significant genetic risk factor for them, yes.
Okay, moving to the immune and endocrine systems, the classic DiGeorge components. Let's reinforce the impact there. T-cell deficiency.
That's a direct result of the underdeveloped thymus, the thymic hypoplasia. If the thymus isn't working properly, T cells can't mature. Without mature T cells, the immune system can't effectively fight off many types of infections, especially certain viruses and fungi. This leads to recurrent, sometimes severe, infections.
And does this immune system problem cause other issues besides just infections?
It can. The sources mention that because the immune system isn't regulated properly, there's also a higher risk of autoimmune problems down the line. Things like immune thrombocytopenia, where the body attacks its own platelets.
Yep.
And also atopic disorders, like severe allergies or asthma. It's a broader immune dysregulation.
And the endocrine side, the hypocalcemia from the parathyroid issue, that needs careful management.
Lifelong vigilance, absolutely. The parathyroids are supposed to sense low calcium and release hormone to correct it. If they're underdeveloped, that system fails. Maintaining calcium isn't just about preventing seizures in infancy, it's crucial for nerve function, muscle function, and bone health throughout life.
Right. Okay, lastly, let's revisit those VCFS-type features. The structural issues like the palate and the developmental aspects. These are often the things noticed later.
Correct. The palate abnormalities are really significant. It can range from that obvious cleft palate to the more hidden submucous cleft. Either way, it affects the muscles needed to close off the back of the nose when speaking.
Leading to that nasal speech quality.
Exactly, hypernasal speech, which can be hard to understand and really impact communication and social development.
And the developmental delay piece. That sets the stage for later issues, too.
Yes, developmental delay is very common. This often translates into learning disabilities as the child gets older. Difficulties with things like abstract thinking, executive function, processing speed, these are characteristic. These cognitive challenges, combined with the facial features and palate issues, were often the clues that led to a VCFS diagnosis before the genetic link was fully understood. #3, molecular basis, and diagnostic certainty.
Okay, so that covers the what. Let's get into the why. We know this deleted segment is pretty big, 1.5 million base pairs with 30 to 40 genes inside. Why such huge variation in how patients present? Why isn't everyone the same?
That's really the million-dollar question, isn't it? The phenotypic heterogeneity. The best explanation we have relates to the fact that the exact start and end points of the deletion can vary slightly from person to person.
The break points aren't always identical.
Precisely. Think of that 1.5 millibare region like a street block with, say, 40 houses, each representing a gene. The deletion might usually take out houses 1 through 40, but sometimes, maybe it's houses 3 through 38 or 1 through 35. The region contains these things called low copy repeats, or LCRs, which are sequences of DNA that look very similar. They make the chromosome prone to errors during meiosis, specifically unequal crossing over where the DNA breaks and rejoins incorrectly.
So depending on exactly where that break happens,
you might lose a slightly different set of genes or keep a crucial one near the edge that someone else loses. If a gene critical for, say, thymus development is lost in one person but spared in another, who loses a gene more critical for heart development, their clinical pictures will diverge.
that makes sense. But among all those deleted genes, science has pinpointed one major player that seems to explain a lot of the core structural problems, right? The TBX1 gene.
Yes, TBX1 is definitely the star player for many of the physical features. It's what we call a TDOX transcription factor.
Transcription factor, so it controls other genes.
Exactly. It doesn't build tissue itself, but it acts like a foreman, binding to DNA and switching other genes on or off at the right time and place during development. It orchestrates the process.
And where TBX1 is active in the embryo lines up perfectly with the affected systems.
It really does. TBX1 is heavily expressed, meaning it's switched on, in the pharyngeal arches and pouches, those embryonic structures we mentioned earlier. And since those structures go on to form the face, the palate, the thymus, and the parathyroid glands.
Losing one copy of the TBX1 foreman messes up the construction of all those things.
That's the link. Losing one functional copy of TBX1 disrupts the developmental signals for all those related structures, leading to the characteristic malformations.
It's not just losing the gene, it's losing its downstream effects. Can you give an example of that cascade?
Sure. A good one involves another gene called PX9. We know PX9 is important for developing the palate, parathyroids, and thymus. Research shows that TBX1 actually targets and regulates PX9.
So TBX1 tells PX9 what to do.
In simple terms, yes. So if you only have one working copy of TBX1, you don't get enough signal to ping AX9. And if PX9 isn't fully activated, the structures it's responsible for, palate, parathyroids, thymus, don't develop properly. It's a clear molecular pathway connecting the master regulator to the specific defects seen in the syndrome.
A beautiful, if devastating example of a developmental pathway gone wrong. #B. establishing the diagnosis. Okay, so given this variability from a baby in cardiac crisis to a child with learning issues, how do doctors actually confirm the diagnosis? Clinical suspicion must be tricky sometimes.
Suspicion is the starting point, but for certainty, especially for genetic counseling and planning long-term care, you need genetic confirmation. And the gold standard test for this specific deletion is fluorescence, in situ hybridization, or FIH.
FAHH, how does that actually let you see the missing piece?
It's quite clever. You use specially designed DNA probes that have fluorescent tags attached so they glow under a specific type of microscope. One probe, maybe tagged green, is designed to stick to a stable part of chromosome 22 that's always there, just to confirm you're looking at the right chromosomes.
Okay, control marker.
Right. Then you use another probe, maybe tagged red, that's designed to bind only to that specific 22q11.2 region that's often deleted. You apply these probes to the patient's cells, usually from a blood sample.
And in someone with the deletion.
In a person without the deletion, you'd see both the green control signal and the red 22q11.2 signal on both copies of chromosome 22. But in someone with the deletion, the green control probe will still light up on both chromosomes. However, the red probe will only find its target sequence on the one healthy chromosome 22. On the other chromosome 22, that segment is missing, so the red probe has nowhere to stick.
So you see green on both, but red only on one.
Exactly. That visual confirmation, one red signal instead of two, proves the deletion is present on one copy of chromosome 22. It gives you diagnostic certainty.
That's incredibly clear, and it works well.
Very well. FIH detects the deletion in something like 90% of cases that fit the classic DiGeorge picture, and around 80% of those fitting the VCFS picture. And importantly, it's sensitive enough to pick up the deletion in that 30% group who only present with the heart defect.
Reinforcing that the deletion is the root cause, whatever the outward symptoms look like.
Precisely. #V. Research focus and long-term advancements.
So if the heart problems and immune issues are often the big challenges early on, it seems like the neurological and psychiatric risks really define the experience for individuals with 22q11.2 Deletion syndrome as they get older. This feels like a critical area of current research.
It absolutely is, and it has implications far beyond just this syndrome. The high rate of psychotic illness is perhaps the most significant long-term burden. #TAG, the high risk of psychotic illness.
Let's talk about schizophrenia specifically. The statistics are pretty alarming compared to the general population risk.
They are genuinely staggering. In the general population, the lifetime risk of developing schizophrenia is about 1%. But for individuals carrying the 22q11.2 deletion, that risk skyrockets. Current estimates suggest that approximately 25% of adults with the syndrome will develop schizophrenia.
1 in 4. That's a 25-fold increased risk.
An enormous increase. It makes this single deletion one of the strongest known genetic risk factors for schizophrenia in humans.
Which is why it's such a focus for research, right? It's a kind of model system.
Exactly. Schizophrenia in the general population is usually complex, polygenic, lots of genes contributing small amounts of risk. But here you have a huge chunk of that risk tied to one specific identifiable deletion. It gives researchers a much clearer target to study.
And if you flip the question around, if you look at people diagnosed with schizophrenia, particularly if it started early, in childhood, how often do you find this deletion?
That's also quite revealing. Studies have found the 22q11.2 deletion in about 2% to 3% of individuals diagnosed with childhood-onset schizophrenia.
Which is way higher than its frequency in the general population.
Massively higher. Finding it in 2–3% of that specific psychiatric group, when it only occurs in 1 in 4,000 births overall, it really hammers home the strength of the causal link between this deletion and psychosis. It's like a natural experiment showing us a major genetic pathway into schizophrenia.
And it's not just schizophrenia, right? Other neurodevelopmental issues are common too?
Yes, definitely. Attention deficit hyperactivity disorder, ADHD, is very common, seen in maybe 30% to 35% of affected children. Anxiety disorders, social interaction difficulties, and specific types of learning disabilities, especially nonverbal ones, are also much more frequent.
So it's a lifelong picture of potential neurological and psychiatric challenges.
It really is. The combination of early developmental delay, the high rate of ADHD, and then this looming 25% risk of psychosis means that comprehensive mental health support and monitoring are absolutely essential throughout the person's life.
This brings us back to the genes. If TBX1 is the main culprit for the physical structural stuff, the heart, the palate, the thymus, it seems unlikely it's also the sole driver of the schizophrenia risk.
That's the critical point, and where current research is intensely focused. The sources are clear on this. While TDX1 explains a lot of the physical malformations linked to those pharyngeal arches, it doesn't seem to fully account for the cognitive profile or the dramatically increased risk of psychiatric illness.
So the answer must lie with some of the other 30-odd genes in that deleted segment.
That's the strong hypothesis. We have this deleted block of 30 to 40 genes. TBX1 accounts for some key features, but there must be other genes within that deleted segment whose loss specifically impacts brain development, neuronal signaling, synaptic plasticity, things that ultimately lead to conditions like schizophrenia or ADHD.
So the hunt is on to find those specific psychosis risk genes within the 22q11.2 deletion.
Precisely. Researchers are looking hard at candidate genes in that region known to be involved in, say, dopamine signaling pathways, which are heavily implicated in psychosis, or genes involved in how neurons migrate and connect during brain development. Identifying which specific gene loss, or maybe combination of gene losses, drives that 25% schizophrenia risk. That's a major goal.
And the implications could be huge.
Huge. Not just for potentially developing targeted treatments for individuals with 22q11.2 Deletion syndrome, but also for potentially uncovering fundamental mechanisms of schizophrenia and other psychiatric disorders that apply to the wider population. #TAG-B, connecting to the bigger picture.
Stepping back for a moment, this whole syndrome is a really powerful illustration of a fundamental genetic principle, isn't it?
Absolutely. It's a textbook case of what happens with gene dosage imbalance. Our bodies are incredibly finely tuned to work with 2 copies of most genes. Losing 1 copy, having only 50% of the normal amount of protein product from 30 or 40 different genes simultaneously is just too much disruption for normal development to proceed smoothly.
The system can't compensate adequately.
It often can't, especially for complex developmental processes involving multiple interacting pathways. It highlights how sensitive development is to having the right amount of these genetic instructions at the right time.
And the clinical take-home message from all this, for healthcare providers, for families.
Vigilance. Comprehensive, lifelong, multidisciplinary vigilance. Because the presentation is so variable, you can't just focus on one aspect. Fixing the heart defect in infancy is critical, obviously, but it's not the end of the story.
You need a team approach that follows the patient over time.
Absolutely. Regular cardiac follow-ups, monitoring calcium levels, checking immune function, providing necessary educational and developmental support, and crucially, proactive screening and support for mental health issues. Knowing about that 25% risk for psychosis means early intervention, psychoeducation for the family, and potentially early treatment if symptoms emerge can make a real difference to long-term outcomes.
It demands a truly holistic view of the patient's health across their entire lifespan.
It really does. Understanding this deep dive informs that lifelong care strategy.
We've covered a huge amount today, journeying from a tiny spot on a chromosome right through to lifelong health implications. Let's try and boil down the absolute core takeaways for everyone listening.
Okay, first, the big one. Chromosome 22q11.2 Deletion syndrome is the single genetic root cause that unified DiGeorge syndrome and velocardiofacial syndrome. There are different manifestations of the same deletion.
Right. Second, the wide range of problems heart, immune, calcium, face, palate makes sense because the deletion affects genes crucial for the development of the embryonic pharyngeal pouches where all these structures originate.
Exactly. And third, while the gene TBX1 within the deleted region explains many of those physical structural problems, it doesn't seem to be the whole story for the significant cognitive differences, and particularly that dramatically increased risk 1 in 4 of developing schizophrenia later in life.
Which leads us neatly to our final provocative thought for you, the listener, to mull over. We know this deleted 1.5 million base pair segment on 22q11.2 contains 30 to 40 genes. We've identified TBX1 as a major player for the structural issues, but what about the other 30-plus genes lost in that deletion? Given the profound psychiatric consequences, like that massive schizophrenia risk, what roles do these other less understood genes play, and how might figuring out their functions research that's happening right now not only lead to better interventions for people with this specific syndrome, but also potentially unlock completely new insights into the biological basis of complex psychiatric conditions like schizophrenia and bipolar disorder for everyone?
It's a key question. This relatively small deletion might hold some incredibly important answers about brain function and mental illness. The breakthroughs could very well start right here, within that 22q11.2 region.
A really powerful place to end. Thank you for joining us for the deep dive.